Good morning, everyone. Today we are going to start with Chapter 2, Transmission of Genes and Genetic Recombination. We are going to discuss genes on chromosomes and DNA with transcription, translation, the processes of protein formation or protein synthesis, and we'll do some recall about genes and alleles. In a cell, we can find the nucleus, the mitochondria, the organelle responsible for the energy, and other parts. The nucleus is the main part. It has the chromosomes. As we've said, in a diploid cell, there are 46 chromosomes, while in a haploid one, there are 23 chromosomes. A chromosome is the major part in the nucleus that constitutes the genetic information. It means like eye color or skin or any other trait in a human being. Okay, so in the chromosome we have the genetic material. How is this genetic material arranged? We have a structure called DNA. DNA or deoxyribonucleic acid. It is the major part that carries the genetic information. It means represented by genes. As we all know, we have genes for every trait in our body. So DNA is present inside the chromosome, as we can see in this structure. It is a double helix. It means it is doubled or like a ladder. In the chromosome, we can find the DNA, which is responsible for all the genetic information and all the genes. A recall on DNA and genes. We've said that DNA or deoxyribonucleic acid, it is the macromolecule or large molecule that has double helix structure, as you can see. So it is composed of two strands. These two strands are composed of many nucleotides. What do we mean by nucleotide? A nucleotide is composed of three components: phosphoric acid, deoxyribose sugar, and a base, nitrogenous base. The nitrogenous bases are four: adenine A, cytosine C, guanine G, and thymine T. So one of these bases is present in every nucleotide. And how are these nucleotides present? We can see here the chemical arrangement. We have two strands that are complementary. G pairs with C and T pairs with A by hydrogen bond. It means this is a base pair, GC or CG and TA or AT. And these are two strands complementary to each other. This is the structure of the DNA. On the chromosome, I have the DNA. It means in every chromatid, I have a double helix DNA. And this DNA represents the genes which code for all the traits in an organism. Every gene has a specific sequence of DNA, as you can see, and every gene is responsible for a specific trait or function in the body. And genes are represented by alleles. An allele is the form of the gene. Okay, it is symbolized by capital if it is dominant and small letter if it is recessive. For example, here I have a pair of chromosome. It is called homologous pair, one from the dad and one from the mom. After DNA replication, I have a homologous duplicated chromosome. On each chromosome, I have allele. Here we have A capital and here we have A small. So for this gene, we have the genotype A capital A small. It means one dominant and one recessive, heterozygous genotype, or a genotype having two different alleles. This means that upon duplication, A here becomes AA on this chromosome, and on this chromosome we have A small because it is duplicated. But we write the genotype as A capital A small. So a homologous chromosome or a homologous pair has the same size, the same position of centromere, and same banding pattern with the same genes, but not necessarily the same alleles. We've said that every gene is a fragment of the chromosome, or it is a part of the DNA. It is responsible for a specific hereditary trait. Now, every gene codes for a protein, but the expression of these proteins is done by two processes, transcription and translation. Transcription first occurs in the nucleus because DNA cannot get out of the nucleus, so we need a mediator. This mediator is mRNA. So DNA must be transformed to mRNA by a process called transcription. How does transcription occur? First, we have two strands of DNA, 5' 3' and 3' 5'. They are complementary. One is called coding or non-transcribed, and the other is called template, which is non-coding or transcribed strand. Now, mRNA will be synthesized by RNA polymerase enzyme. It is complementary to the strand that is the template or transcribed. So the complement of C in the RNA is a G, and the complement of A is U, because in RNA we don't have T, we have U or uracil. So the complement of C is a G and the complement of A is U, with the complement of T is A. So we have C with pairs with G, A with U, C with G, A with U, and T with A, and we continue like this. So this mRNA strand is complementary to the transcribed or template strand, but it is the same or identical to the coding or non-transcribed, but instead of T we put U. This is transcription that occurs in the nucleus and produces mRNA. Now translation, it is the second process of protein synthesis. It occurs in the cytoplasm. Now mRNA is in the cytoplasm. It will be transformed into a protein or a sequence of amino acids. According to the mRNA sequence, amino acid will be joined and formed in a sequence. For translation, we need a ribosome, which is composed of two subunits, a small subunit and a large one. The large has three sites, E, P, and A sites. Also, we need a tRNA that has two sites, one for the anticodon, which is complementary to the codon present on the mRNA, and that will bring the correct amino acid that has a site on the tRNA. This amino acid is specific for the codon present on the mRNA. Initiation step starts first with the amino acid methionine, which has in the tRNA the anticodon for the codon on the mRNA AUG, which has complementary UAC. Okay, so this tRNA brings UAC anticodon and the amino acid methionine. It is present in the P site, and the ribosome carries this tRNA to the mRNA sequence. This is the initiator. Then we have elongation. Another tRNA comes with an anticodon complementary to the codon present on the mRNA and carrying the specific amino acid. Then the amino acid, which is methionine on the first tRNA, binds with the amino acid on the second tRNA, which is present in the A site. So a peptide bond is formed between the two amino acids, and this tRNA, which first carries the methionine, will be detached. Then in the elongation step, the ribosome moves by one codon. Thus, this amino acid, this tRNA becomes in the E site, and the second tRNA becomes in the P site, and the A site becomes empty. Now, another tRNA brings another amino acid specific for the codon present in the mRNA, and the initiator, the one which carries the first amino acid, which is methionine, now gets out. Okay, and then this tRNA sits in the A site, and we continue the process of elongation to reach the termination where a stop codon will be present, because we have a codon on the amino acid which is specific for an anticodon that has the stop 1, it means the mRNA has finished and the process of translation is now finished. So we have a sequence of amino acids that will detach, and it is formed by peptide bonds. The other components will detach and the amino acid sequence is now ready and the protein is synthesized. As a summary, the DNA, which has the double helix structure, which is present on the chromosomes, and the gene is a part of the chromosome or a part of the DNA, it is transcribed into mRNA. The mRNA gets out of the nucleus, then it is translated into protein or sequence of amino acids, and the amino acids are present in the cytoplasm. This process of translation occurs in the cytoplasm. And this is a review for transcription and translation. For example, here I've took a part of the DNA sequence. The mRNA is complementary to the template strand or the transcribed one, and this transcript or mRNA forms a protein by process called translation. First it starts with methionine, then it continues with the sequence to reach the stop codon. This is the protein that has its specific function, and it's important to note that all our body cells carry the same genes or the same genetic information, but these genes are not equally expressed. For example, the gene coding for insulin is found in all the cells, in all our body cells, but it is expressed only in pancreatic cell. It means every gene is expressed in the place where it functions, but all genes are present in our cells.